Hello everyone, and welcome to my third video essay for exam number 3. This one we're going to be talking about blood, everything about blood, the composition of blood, and how it can clot and how it works, and all sorts of things about blood. So let's go ahead and get started. If we take a sample of blood from a patient and put it in a vial, we'll have our blood here in our vial. And then if we centrifuge it, so we spin it around really fast, we'll have the heavier portion of the blood settle to the bottom, which is the red blood cells. So if you can imagine, these are just the red blood cells. And then we'll have a medium layer that will be white, and that will be the white blood cells. I'll just represent it, white blood cells like that. And then we'll have this clear liquid portion at the top of our blood. Now, for healthy individuals, our percentage of red blood cells that make up our total blood composition is going to be around 40%. So it'll be anywhere from like 30 to 40%, and that can vary based off of your age as well as where you live. So if you're in a higher elevation, that'll be a higher amount or higher percentage. It also gender affects it. So if you're a male, males tend to have a higher blood count, or hemocrit is the correct word for it. And so there's a couple of factors, but that's essentially the composition of blood. And there's some really important things that we find in all parts of it, but let's talk about plasmid for a second. So plasmid. If you're a poor college student like me, you've probably donated plasmid before for money. So plasmid, why is it important? Why do we donate it sometimes? Well, there's some very important things in it, including proteins and antibodies. So it's an important part of our immune system. And then there's a lot of water and solutes. Solutes meaning like salt, maybe some potassium, maybe some ammonia, maybe some things that need to be filtered out of the blood in the kidneys. So I'll just bunch those two things together, water and solutes. But the real important things that we're going to be talking about today are the proteins and the antibodies that are found inside of plasmid. So one of the most common, in fact it is the most common protein that's found inside of our blood and in our body, is called albumin. So albumin is a very prevalent protein found in your blood, and it has a really important function when it comes to helping with recycling red blood cells. Red blood cells are constantly being recycled in our body. The average lifespan of a red blood cell is usually around 120 days, and every second we create, give or take a couple thousand, we create about 2 million new red blood cells. That's a lot of new red blood cells every single second of every single day. And we get rid of our old red blood cells, the ones that have spent those 120 days, almost just as quickly. And so what happens when we get rid of red blood cells? What does that look like? Well, let's talk about it. So our red blood cells will be broken down, or lysed is the technical term for it. So inside of our red blood cells, we have a bunch of things. We have some proteins, we have some, the phospholipid bilayer that makes up the membrane. We have some DNA. Well, actually red blood cells don't have a nucleus, but that's besides the point. Anyways, when we break open, or when we lyse our red blood cell, we have all sorts of things that come out, like potassium, maybe some sodium, calcium, chloride, a lot of solutes to get recycled when we lyse our old blood cells. The phospholipid bilayer goes back to be recycled. Our body is really good at reusing the resources that it already has inside of it. But one thing that it can't recycle is something called hemoglobin. I'll write it. Hemoglobin by itself is actually a toxin. So when it's inside of our red blood cells, hemoglobin is actually the substance or the protein that is responsible for latching on to oxygen, molecular oxygen, and then that's how our blood is able to be oxygenated and carry oxygen to tissues that will need that for aerobic respiration. Now, when we lyse a red blood cell, we just have hemoglobin by itself. And unfortunately, it cannot just stay that way. Otherwise, it will create some problems for us. So our body needs to figure out how to get rid of hemoglobin in a safe way that doesn't leave us feeling sick. So what happens is we will have hemoglobin being picked up by that protein that we talked about, albumin. Albumin carries hemoglobin, I'll just put Hg for short, over, and it will be carried over to the liver, I believe. Yeah, to the liver. So that hemoglobin or that heme will be picked up by albumin, carried over to the liver. Draw a little liver there. And then from there, what our liver does is it attaches a bunch of glucose molecules all over that hemoglobin. So to give us a visual, I didn't put an R on it. So what happens is, say this is our molecule right there, hemoglobin. Now it's over here, and the liver will attach all these glucose molecules to it. I'll just do a bunch of G's. And so it's kind of just like completely covered in sugar or glucose. And then from there, it gets sent out through the bile duct into the intestines. And inside of the intestines are like microbiome in there. So all the little bacteria will start eating the outside of this hemoglobin. And, it gets processed so that it's no longer hemoglobin. It becomes something called bilirubin. So bilirubin. I'll write that down for you. So it becomes hemoglobin, gets transferred and turned into bilirubin, which is basically just like a processed version of hemoglobin. And then that bilirubin becomes attached to a bunch of glucoses. We call this process conjugated. So we call it conjugated bilirubin. And that just means it has a bunch of glucose on it. And then from there, that's sent into the intestines where it will be started to be eaten by the bacteria who love glucose. They'll start eating, snipping off all these glucose molecules. And from there, it will be transferred up into our bloodstream from the intestines. And from the bloodstream, it'll be filtered through the kidneys. And from the kidneys, it'll be processed another round of processing. So it will no longer be bilirubin, it will become urobilin, which is something that we are able to pee out. The reason why it has to become urobilin is because bilirubin is a lipid-soluble substance, meaning it does not become a solute inside of water. It kind of keeps to itself. Kind of like if you've ever tried mixing oil and water together, it doesn't work very well. And even if you're able to get it by mixing it really fast, it'll start to separate as soon as you stop doing that. And that's what bilirubin does inside of our body, and that's one of the reasons why we have to get rid of it, because it is not a healthy substance to stay inside of our body. So once it becomes water soluble, it becomes urobilin. That urobilin then goes through the urethra, and we're able to pee it out through the urinary tract. And that's how we get rid of, ultimately, hemoglobin, which is a kind of a side effect of lysing our old red blood cells. Now, why is this important? Well, if you've ever heard of something called jaundice, then you are familiar with one of the most famous symptoms, which is basically you just turn yellow. And what that does, why that happens, is because bilirubin is yellow in complexion. If you got a bunch of bilirubin put together and put it on a plate, you just see a big splotch of yellow. And so, because bilirubin is only fat soluble, it's not water soluble, if it's not processed properly and it doesn't exit the body, then it'll start to get stuck in our tissues. It'll be stuck, in our skin, in our eyes, in our organs. It'll just basically kind of build up everywhere around the body, and that's where the yellow complexion comes from. Now, there's two types of jaundice that you can have. Two main types. There's the type where you have the conjugated bilirubin that's flowing through your blood, that's causing that yellow, or you can have the unconjugated version of bilirubin. So the one that just came from the albumin and into the liver. And if it's a conjugated bilirubin, then you know that it's a problem. The problem is not with the liver, because the liver's job was attaching those glucose. So if it's already conjugated, then we know the liver was doing its job. But if it's found in the blood instead of in the intestines where it was supposed to be, then it's an obstruction of the bile duct. Because remember, that's the pathway we take to go from the liver through the bile duct into the intestines, where that bacteria begins to kind of break it down a little bit because of those glucose molecules. So if we find glucose covered bilirubin in the blood, we know it's not a function of the liver that's wrong, it's the bile duct, and that requires a surgical treatment. If we find just normal bilirubin just going through our blood everywhere, then we do know then that's a problem with our liver. Our liver is not functioning properly, it's not attaching or conjugating with glucose, our bilirubin, like it should. And that's where we get treatments where we're able to, we don't necessarily need surgery. In extreme cases, you might need something like a liver transplant, but usually the doctors will have different treatments that aren't as invasive as surgery for that. So that's why it's important that our body filters out bilirubin, or as it originally was, our hemoglobin, or our hemes, from our red blood cells that get lysed. Perfect. All right. Next, we will be talking about hemostasis, which is basically how we're able to kind of control blood flow even when we end up having a hemorrhage. So if you've ever cut yourself, or you've ever had a scrape or a scab, you'll know that your blood doesn't just continually flows out of you forever, otherwise you would die. Eventually you have a blood clot which forms a scab. Let's talk about the process of how blood clots form and the cascade process that goes with that. So I'm going to draw a blood vessel for us here. I'm going to draw the blood vessel in black, and then I'll draw the red blood cells and other components in red. So we have our blood vessel. I'm drawing it very blown up here, very magnified. And then on the inside of our blood vessels, we have a single layer of epithelial cells. So I'll just draw them like that, all on the inside, lining inside of our blood vessels. And they all sit on top of this protein wall made up of collagen. So this collagen wall I'll draw as a squiggly line above our epithelial line, like that. So we got our epithelial cells in our collagen wall. And as a part of the components of our blood, we have our red blood cells that are just flowing through. But then we also have something known as platelets. I'll draw them in green so we can see these here. And they kind of look like just little grains of rice. Just like that. And basically what these are they're the very important part of our cascade that creates clots. They all have these receptors on them. That are meant to be stimulated to activate the cascade that will eventually lead to a blood clot. So I drew them as kind of like tunnels, kind of ugly looking things. Obviously none of this is really to scale, but we'll do our best here. So what happens when we have these channels? Well, first of all, this channel is called the glycoprotein 1V, or the VWFR receptors. VWFR. It has a couple different names. It can also be called the GPIB. But basically, we have these platelets that have an activation site on these receptors, and usually when our epithelial cells make a ligand that attaches to these receptors, that ligand is known as von Wunderbar factor, or just VWF for short. VWF. They're just like little ligands that are made by our epithelial cells, and when they attach to the cells, or to the receptors on our platelets, they don't do anything. So basically, they're inhibitory, and our epithelial cells are constantly making this. And if you think about this, this is what you want for your body, right? Because if you have constant blood clots that are happening just spontaneously inside of your blood vessels, then you'll always have very high risk of having a stroke anywhere, right? Because you get that cascade that goes on. You have blockage of blood, and that can become super dangerous super fast. So this VWF ligand just attaches to all of these receptors on the platelets and kind of just keeps them chill. It keeps them from spontaneously going off and starting a cascade. Now, say you get a cut. Say, you were running on the sidewalk and then you tripped and fell and you scraped your knee. Now, if you have kind of a deeper scrape, you'll have more blood, obviously. But what happens is when you scrape right here, you'll have a breaking of your blood vessel, and then you'll have some of these platelets will begin to rush out along with red blood cells and everything else that makes up our blood, and it will come into contact with the collagen wall, with that protein wall that's kind of sitting on the outside of our blood vessels. Now, this is when we get a cascade that begins. So I'll draw a platelet that kind of connects to the collagen. And once this happens, it'll begin to activate. So platelets are kind of interesting. If you've ever seen like the movie Alien, they kind of look like a little alien that starts to just wiggle and come alive, and it grows really big. It'll grow these big kind of like spider octopus looking arms. kind of overgrow like that. And that's the very beginning of the cascade that creates our clots for us so that we don't bleed out and die when we have a small scrape. When this happens, we get other platelets that connect to our first one that got activated, and they will act very similar to the receptor that is activated by the collagen is also activated by an activated version of a platelet. So one of these like monster spider-looking arms. If we have a platelet that finds its receptor connecting to that, then it will begin to activate as well, and it will grow its own spider arms as well. And then in addition to that, we have these channels that will grow in between activated platelets. Drew one right here. And these channels are known as fibrinogen. Fibrinogen will act as a bridge to hold together the platelets, and it kind of is known as the platelet plug. It just is a tight junction that holds it all together, and eventually the idea is you get so many of these platelets that are plugging up and activated like this that it completely obstructs any blood flow to flow out of the blood vessel, which is good. That's what it's designed for. But there's a couple of things that can go wrong. So we just kind of went through what we went through is the ideal scenario where you get a cut and then your platelets activate as they should and they keep the blood inside of the vessel, and then that way you can just keep on your merry way and you don't even hardly notice until the scab starts itching, which, by the way, it starts itching because you have certain immune cells that are surrounding the damaged area where the scab is that will release histamine, which is kind of an irritant, inflammatory, and you'll want to scratch it. And then you do end up scratching it, and eventually that becomes a new layer of skin or cells. So let's talk about what if something went wrong with our blood vessel. What if we had our blood vessel here, we have our epithelial cells here, and our collagen wall surrounding it. What if we had a condition where some of these epithelial cells started to die off? Say we have 2 or 3 in a row that died and just kind of weren't there anymore. This actually is a symptom, or it could be an effect of long-term tobacco use. So if you smoke a lot, stop. It's not good for your health. But also, this was a reason why. So you see some of the epithelial cells will just die off and they won't grow back due to the toxins in tobacco. But when that happens, we get exposed collagen wall. Now, we just talked about what happens when you have a platelet that comes into contact with that collagen. Well, you can probably guess what would happen, right? You have a platelet that spontaneously attaches that receptor to the collagen, and then it begins to activate. And that creates a cascade where it begins to activate another one, and then another one, and then another one. And eventually, it snowballs into this huge clot. And then that clot can possibly break off of that collagen wall as the pressure on the arterial side builds and builds until it pushes it downstream, and then it gets lodged in another place, and that's where you get your strokes. And this can be extremely dangerous. And so there's a couple of treatments that doctors have for individuals who are at higher risk for this to happen. And there's 2 main pathways that this can happen. this pathway that we talked about right here is known as the intrinsic pathway. Intrinsic because it is activated from the thing inside of the blood vessel. Intrinsic pathway. So I'm going to write down a lot of Roman numerals, and then I'll explain it all. But basically, intrinsic pathway is when you have something called factor 12 that hits against the collagen wall and turns it into factor 12A, which in turn differentiates into factor 10 and factor 9A. And then factor 9A, in addition to factor 8, will then create factor 10A plus factor 5. Factor 10A will then create factor 2, or it will turn on a cascade of factor 2, as well as factor 2A. Factor 2A then activates factor 1, as well as factor 1A, and finally factor 13. Alright, let's kind of go through this. I'm not going to explain every single little thing here, but the idea is we have a single action that occurs. Factor 12 coming into contact with our collagen wall. A single one of these will then activate, and it will become factor 12A, and it will activate thousands of factor 10 and factor 9A. And then thousands of factor 9A plus thousands of factor 13 will create, or will activate, even more thousands of factor 10A, which in turn will activate even more thousands of factor 2A, which will then create or activate thousands of factor 1A. So if we multiply each by the power of 1,000, we get down to here, we're talking about millions and millions of factors that have become activated. That's how we can get really good at clotting cuts and injuries where we'll have lots of blood loss because we have this response system. And that's, again, this is one pathway known as the intrinsic pathway. There's one other major pathway that we want to talk about, and that's something called the extrinsic pathway. You could probably guess that involves something from the outside of the blood vessel initiating a cascade similar to the intrinsic pathway. So extrinsic pathway occurs when we have factor 3 being released by cells outside of our blood vessel. I'm not really going to get into what these factors are exactly and how they function, what their mechanisms are, because to tell you the truth, I'm not entirely sure. I am still just an undergrad student, and science has not progressed to the point where undergrad students need to know that yet. I mean, if they do, that's really cool, because it's tough stuff. Anyways, so we have any cell. Pick a cell. You could choose a muscle cell, smooth or skeletal muscle. You can have, maybe some lipid, like fat cells. All cells will create factor 3 when you have some sort of trauma that occurs. And factor 3 will act very similar to collagen where it will activate anything. It will activate a specific thing. It will activate pathway and does not block this. It will activate something called factor 7. So if somehow factor 3 is able to permeate our blood vessel wall, it will activate factor 7. And then from factor 7, it will activate factor 10 and 10A. And then from there, it's the same as the intrinsic pathway. So that's really the only difference between extrinsic and intrinsic pathway, is the starting steps. Now you can imagine if you want to stop one of these cascades, you would want to start at the very top, the thing that begins the cascade. So for intrinsic pathway, that would be factor 12, right? And for extrinsic pathway, that would be factor 7, right? If there's no factor 7, then factor 3 cannot start that intrinsic pathway, no matter how much of factor 3 there might be, if that makes sense. Now I'm going to talk about two different drugs that are known as blood thinners, but what they actually do is they inhibit or they stop either factor 12 or factor 7. And both of them just do one or the other. And so usually doctors will prescribe one of these. They will never prescribe both because that means you will have no ability to clot at all and then you will die. So let's talk about which one is which. So the drug that inhibits the intrinsic pathway is known as heparin. I think I'm pronouncing that right. But heparin will basically just totally destroy all factor 12s. So if you give a patient heparin, then you can bet that if they have any clotting, it is not due to the intrinsic pathway. That intrinsic pathway is essentially shut down, which is good for keeping high-risk patients from having internal blood clots. And then in order to shut down extrinsic pathway, you need something called coumadin, I think is how you say it, coumadin. Go ahead and circle that in red as well. Coumadin will shut down all factor 7s so that when a factor 3 comes into your blood vessel, you have no cascade initiated. So these are two really important medications to keep in mind. If you already have been administered one, you never want to have the other as well, because the two combined is a lethal combination. All right, that is it for this week. Thank you for watching, and see you next week.